Hello, this is our presentation on the influenza virus and why there is no universal vaccine. By me, Wasim Musleh.
Tonya Garcia.
Ezekiel Mendez.
Dorian Padilla.
Marsha Lee.
And Kyle Hermesera.
There are many misconceptions on vaccines and their effectiveness that rose up during the COVID-19 pandemic. The flu vaccine faces these similar, if not exact, debates and misconceptions. As a result, these issues and misconceptions have been brought onto a political stage. Our goal is to educate as many individuals as possible about vaccines and the properties that render them effective. For example, there is a false understanding that vaccines trigger an immunological response that will result in complete immunity against this pathogen, when in reality, a virus is an organism that is rapidly mutating and adjusting to its changing environment and will overcome these barriers established by the vaccine. As a result, a new strain of the virus may arise with these mutations incorporated within the genome. Flu vaccines are created based on heavy analysis and studies conducted by scientists to determine which strain will become the dominant strain during the flu season. By vaccinating as many individuals as possible prior to the flu season, the amount of individuals that get sick and spread the virus is decreased. Our research is based on how vaccines train the immune system and why there is no universal vaccine for the influenza virus.
I'll be talking about the background. As we're all aware, the flu is a viral infection that attacks the respiratory system. Common symptoms include fever, chills, aching muscles, and etc. In order to reduce the chances of getting the flu, individuals should receive a yearly flu vaccine. It is important to get vaccinated every year since the flu virus evolves very rapidly, meaning last year's vaccine may not protect you from this year's virus. The Spanish flu pandemic, which occurred in 1918, was the deadliest in history. It affected 1/3 of the world's population and killed up to 50 million people. The flu had a major impact on the livelihood of individuals throughout the years. We'll now dive deeper into the technological methods used to create the flu vaccine. Currently, the FDA has 3 technological methods approved for the manufacturing process. The 3 different types include egg-based, cell-based, and recombinant. The egg-based flu vaccine was the most common method. However, currently the most common would be the recombinant flu vaccine. Since many individuals are allergic to egg products, this was a major setback for the egg-based flu vaccine. For the egg-based vaccine, pharmaceutical companies will get a list of candidate vaccine viruses, which are also called CVVs, from the CDC. With these CVVs, researchers inject it into eggs, allow it to incubate, which causes the viruses to replicate. After it replicates, the fluid in the egg will be harvested, killed, then purified. The egg-based technological method can be seen in Figure 1. When it comes to recombinant flu vaccine, the main difference between all the other technological methods is that it does not require the use of CVVs. Instead, it is created synthetically. When creating the recombinant flu vaccines, researchers obtained the hemagglutinin gene. Since hemagglutinin is an antigen, it triggers the human immune system to create antibodies that specifically target the virus. This gene will then be combined with a baculovirus, which will then transport the genetic instructions for making the flu virus hemagglutinin antigen into a host cell. The antigen will then replicate and be purified. There are 4 types of influenza viruses, which include A, B, C, and D. It is important to note that flu A and B are the ones that cause the seasonal epidemics. The influenza virus is composed of both RNA and protein. Every influenza virus has 8 segments that correspond to the virus's 8 total genetic segments. As the influenza virus changes every season due to antigenic drift or antigenic shift, it is important to prevent the spread of the influenza virus.
When looking at the methodology of making this universal vaccine, scientists have emphasized that the best mode of action to make a vaccine with our current technology is by targeting the most conserved regions of the flu virus, as these regions do not have a high mutation rate, and those regions are the hemagglutinin spike protein, neuraminidase spike protein, and the M2 ion channel. The hemagglutinin and neuraminidase spike proteins are essential for the flu virus attachment entry to the host cell, while the M2 ion channel is important for the virus to take in protons to help disassemble the capsid of the flu virus and allow the release of its genetic material into the host cell. As a result, by hindering these conserved regions, it can stop the spread of the influenza virus permanently. And so scientists describe 3 possible approaches in making a universal vaccine, which are the recombinant stock-specific hemagglutinin vaccine, chimeric recombinant hemagglutinin vaccine, and recombinant M2 vaccine. When looking at the recombinant stock-specific hemagglutinin and chimeric recombinant hemagglutinin methods, these are tactics for our immune cells to create antibodies to the stock region of the flu virus as we can see in figure 3, as usually this region is conserved through the majority of influenza A strains. As a result, a vaccine targeting this region can allow for our B cells to recognize this region and create antibodies to target them and stop the attachment and entry of the virus. However, there are still problems that researchers are looking to overcome with these methods that are presented in Table 1, where for recombinant stock-specific hemagglutinin, there's a deletion of the globular head, which can change the hemagglutinin structure. And by also deleting the head, antibodies will not have accessibility to many epitopes to bind onto the virus. And we can see in figure 3 that antibodies are more easily able to bind onto the head region compared to the stalk region. Then for the chimeric recombinant hemagglutinin vaccine, the challenge is being able to make stalk-specific antibodies that can efficiently bind onto the stalk, as seen in figure 3, where there is less ability of antibody being able to bind onto the stalk region. For M2 ion channel vaccine, the goal is to create a vaccine that allows our B cells to create antibodies for the structure, but the main problem associated with this technique is being able to get our antibodies to have access to this region to bind onto. As we can see in figure 2, the M2 ion channel is much more smaller than the other two spike proteins, and we have to keep in mind on how crowded the capsid is with other proteins.
The pharmaceutical company BeyondVax was able to create a recombinant HA universal flu vaccine that was able to progress 3 phases of the clinical trials due to its ability to stimulate an immune response. This trial included about 12,000 individuals from 2018 to 2020, all above the age of 50. But unfortunately, this vaccine was not able to pass that phase due to it not being able to demonstrate a significant difference between the vaccinated group and the placebo groups in the terms of illness and severity.
Another pharmaceutical company, Novavax Incorporated, has been in development of its own universal combination vaccine targeting the 4 most common influenza strains, going through multiple trials with it recently reaching phase 3 of its clinical trial around September 2021. The recombinant quadrivalent influenza vaccine candidate with Matrix M has been shown to produce significantly enhanced humoral and cellular immune responses versus other in-circulation influenza vaccines in older adults. The vaccine itself is composed of recombinant HA proteins similar to what is found in wild-type circulating virus HA sequences, with the addition of Novavax's saponin-based Matrix M adjuvant, in order to further boost immune responses. NanoFlu is also a means of paving the way for another one of Novavax's in-trial vaccines, which target both the influenza and COVID-19 virus.
So after hearing our presentation on the influenza virus, one should have pulled out the following: the basic influenza and vaccine biology, as well as the influenza technology used for the virus. To elaborate, you should now be able to understand why the influenza virus belongs to the Orthomyxovidae family, a negative-sense single-stranded RNA virus, and comes in many variants, as well as the importance of the hemagglutinin HA and neuraminidase NA, also known as the spike proteins. These regions of the virus tend to be conserved, but when mutated, the vaccine efficiency of prior years are lowered, if not repressed, to new mutated viruses. Speaking of vaccines, you should have learned the current vaccine used to combat influenza pandemics, which is the quadrivalent vaccine that cultures viruses using mammal cells, and the importance of this vaccine to prior vaccines like that of the traditional chicken eggs vaccine or other failed vaccines of the influenza virus. Above this, knowing the biology behind the virus, you should now have a broader understanding of how certain detection methods like RT-PCR work when detecting if a person is infected with the virus. To wrap up, although much groundbreaking research has been achieved, many questions remain unanswered. Communication, for example, continues to plague many researchers in terms of how do we better inform the public over viral progressions without causing paranoia or spread of misinformation, which tends to be more detrimental than the virus itself. Finally, even though for many a universal vaccine remains an illusion, emerging vaccine tech like that of the mRNA vaccine tech can also only make us wonder, can the applications of such technology get us closer to a universal vaccine? Thank you for your time. I hope you have learned more on the influenza virus.
And to access the references of our poster, simply press the hyperlink and it will take you to a page of our references.